Right. So this will be the last talk of the of our meeting, of our workshop. Um, uh, this is also serves, I think, as an NITMB seminar and last plenary. It's a pleasure to have Kyoung here. So Jay got his Ph.D. from the University of Michigan in 2013, then went to the MBI where he worked. I think he set the record for the number of collaborators that he worked with at the time. It's 5 or 6, I don't remember.
Yeah.
And was very productive. Went back to Korea, where he became a professor at KAIST. Where
We need to remove this. Yeah.
Please remove it. And also served as the biomedical mathematics group leader at the IBS for 5 years or 6 years. There's really numerous accomplishments. Let me just mention that his software is currently used to track the sleep cycle and the new Samsung watch. That's kind of the latest achievement among many. Jay.
Okay. Hello, everyone. First, I'd like to thank the Creso and Lindsay for organizing this exciting workshop. I'm happy to be involved in this exciting event. So today, I'm going to talk about how cell maintain function robustly even when environmental change or noise. That's the answer I want to pursue so far. Question, yes. So let me introduce some very simple case. So this morning we came to this place and we had some breakfast. Then some food getting into our body, then glucose level will goes up, of course. And then we need to adapt the glucose level to reduce the glucose level. So we call this is adaptation, but this is not enough because the adapted level is not the same with the original level. Then we will die, basically. So that we want to adapt the system to the original system before the perturbation. We call this is robust, perfect adaptation. So then how can we achieve this RPA or robust perfect adaptation? So for instance, this is an E. coli metabolic network. Here, let's change this number 5. So let's increase in number 5 reaction and let's see how the all other fluxes changes. And if you look at it, even we increasing the K5 by 3 times, but all fluxes in this network change initially, but coming back to the exactly original level. So there is RPA. However, if we perturb the K2, another step, you can see that we fail in achieving the RPA. So this is not a trivial phenomenon. Then what is the difference between this K2 and K5? If we can write down the ODE, it's a very easy undergraduate problem. So if we can write down the ODE model, we can derive the steady state. If the steady state, we can just need to check the steady state depending on the K2, but not the K5. So it's a first year undergraduate math problem if we can write down the ODE. However, what if we have only know the network information? We don't know the model. We don't know the parameter, etc. We don't know the equation, then what we can do. So for this, we need to introduce very simple network terminology. So in this biochemical reaction network, V1, V2, V3, V4 represent the species, like molecules. And then E1, E2, E3, E4, E5 represent the reaction, okay? So we first need to identify the conserved quantity in this network. So conserved quantity means, so let's say our initial condition of 4 species is 1, 2, 3, 4. But if the first reaction happened, then first species will be increasing by 1, right? And then if the second reaction happened, then second one will increasing. 1 is switched to the 2, so that decreasing by 1 and then increasing by 1. And then E4 reaction happened, then V1, V2 are changed to the V3 and V4, so that these two are decreasing by 1 and these are increasing by 1, and then E5 similar. Then across all this reaction, we need to find what is the conserved quantity. So you can see that conserved quantity is V3 minus V4. Whatever reaction happened, the difference between V3 and V4 never changed. So this is the first thing we need to know. And then second, we need to check just two conditions. Let's select a certain subnetwork. Let me call gamma. In this subnetwork, we need two conditions. Number one, if there are some species, like a V3 and V4, then all the reactions which have a V3, V4 as a source or reactants should be inside of this subnetwork. So E5 is the only reaction which has a V3 and V4 are reactants, so that E5s are inside of this gamma. So first conditions are done. And then second condition is we need to do elementary math, school math. We need to count the number of the species. V3 and V4 are 2. Good. And then how many reaction? 1. And then how many conserved quantity? 1. So that minus 2 plus 1 plus 1, then we call this is influence index. And the index is 0. If these two conditions are satisfied, then we call this subnetwork a strong buffering structure. And what we proved was, if we perturb anything inside of this strong buffering structure, then whole network fluxes can achieve the RPA. So, and please note that this strong buffering structure solely depending on the network structure, no equation and no parameter thing, right? We only use this network structure. So, for instance, if we perturb the K5 inside of strong buffering structure, you can see the perfect adaptation happen. But if we perturb outside of this subnetwork, then we fail. So, and please note that this is independent of the kinetic parameters or equation we describe this network. So then if we use this concept, here all pink color are strong buffering structure, which satisfy the condition. So then we can predict that if we perturb the inside of this pink, then nothing will happen. But if we perturb black, then something happen. And indeed, this is experimental data. If we perturb this K5, the strong buffering structure, there's no statistically significant change. So this is statistically meaningful bar. But if we perturb outside of the strong buffering structure, you can see that really the flux of this metabolic network changes. And this is another system. This is plant metabolic network. And again, pink is strong buffering structure. So if we perturb the pink, then again, come back to the origin. But if we perturb the black, then we lose the original steady state. And here is the experiment. So if let's change this step, then what happens is if we decreasing this step, there is no reduced metabolic flux, so that plants grow very slowly. But if we increasing this step, plant grow faster. Okay. However, if we perturb this pink, then nothing really happened. So this was quite long-standing mystery in plant, because if you look at it, this one looks same, but why only this one? Huge effect on the plant life was mystery, but the only difference is other is just chain, but this one has this one outside flow, that make a huge differences. So this served quite a long-standing mystery in the plant metabolic field. So this idea of strong buffering structure actually inspired by the prior work of Achis-Mochiski by Kyoto University. Although we're focusing on the flux of the metabolic network, but his work is mainly focusing on the steady state. So if you have an interest in similar thing, please go over his work. Then, so far, I talk about the deterministic system, but our workshop is about the stochastic system. So let's think about this stochastic system. Can we control the stochastic system? So here is a very simple system. X1 is sort of generated, and then it triggers the production of X2. Very simple system. And I want to control the level of the X2. That's my goal. But what I mean is, control means even I change the parameter inside of this cell, still X2 level should be maintained. That's what I mean, the control. So, and the famous controller for this system is antistatic integral feedback loop, or AIF, mainly developed by Kamashi group. So if we add this control, what happens is, one intuition is this. If X2 level is somehow increasing due to the parameter change, then this G2 level will be increased because G2 and G1 bind and then they die together. If G2 level increasing, G1 level will decreasing, and then X1 level will be decreasing, so that X2 level going down. There is some negative feedback loop there, but this is not enough, right? We know that X2 level will be decreasing, but we are not sure it will really go to the original level or not. That's the point. But the solution is very simple. If we don't have this controller, we can drive this mean of the X2 steady state. That is just this simple undergraduate math again. But what you can see is their steady state depending on the original system parameter, right? That means if we change the parameter, their level will keep changing. So we cannot control. However, if we add this controller, then steady state of X2 is mu1 over theta 1. And look at where is mu1 and theta 1. They are outside of this gray box. So that the steady state suddenly don't depend at all inside of its own system. So the key point is original steady state is depending on the original system parameter, but if we add a controller, then it's independent to the original parameter. So here is simulation. With the original gray box, if we increasing the K, then of course X2 level increasing by A-fold. But if we add if we add a controller, then mean level never change about inside of the parameter change. So this, we can achieve again, robust perfect adaptation with this controller. So this was quite a famous concept in this field, but there was a caveat of this controller. So, okay, mean level is well controlled. What else you want to control in stochastic system? Yeah, noise level, right? So the problem is noise is amplified. Although mean is controlled well, but if we use this controller, noise is then why this is bad in the biology is, let's say we have 10 cells, and our goal is we want to maintain this protein level as 10 because we don't want to die, okay? So population mean is very well controlled with this controller. But due to the noise, if you look at the single cell level, jumping, right? We don't want, because we don't want to die. That's the point. So that although this is theoretically beautiful thing, but in real life, in real biology, somehow, yeah, this is recorded. So yeah, use something, yeah. So this is not perfectly good for the biological system. So we want to control this noise as well. So of course, this is not our first thought. There are several studies which try to reduce the noise level. But as you can see, all of this study can reduce it, but not the perfect. So that we want to overcome this problem. So that let's think about how we can attack this problem, how we can control the noise level. Key idea is very simple. How we control the mean level is we control the first moment, right? We just make the first moment depending on the parameter outside of the original system. That's the key idea. Then how to control the noise? Not the first moment we need. Second moment, which should be depending on the outside of the original parameter. That's all. So that how to then do is to make the second moment is controllable. we need this homodimerization or bimolecular reaction. Because if we have a bimolecular reaction, suddenly we have the second order term appear, then somehow you can imagine that second moment is appeared finally. So then if we add this green thing, what happens is now second moment or Fano factor now solely depending on the outside of original system. So, and is it really working? So this is original mean controller. You can see the noise is blow up, increasing a lot. But if we add this green noise controller, not only the mean, we can get a noise level maintained as original system. There is no noise level change. So for instance, this is one simulation. This is original simulation. If we add a mean controller, mean is maintained, but you can see the noise is amplified. But if we add a noise controller now, we can maintain the original noise level as well. Furthermore, actually, we can do further. We can now reduce it because Fano factor is this. So how? Just decreasing the mu2, then we can reduce the noise level as well. So indeed, if we target the noise level, we can reduce further and further, but we failed here. We found that a hard limit is 1. We can reduce up to Fano factor 1, but if we try to more than that, we found that system is blow up, and we don't know why. Yeah, so hopefully someone can solve this problem. Why this limit is Fano factor equal to the 1. Yeah. So this mean controller was actually achieved in the cell using the synthetic biology, which is published in Nature a couple of years ago. Then can we make the noise controller as well? So we made some design for this, but we discussed with some synthetic biologists, but they say with the current technique, it's impossible to achieve this. So someday one can achieve this in the inside of cell, then we can really control the cell very well. So now let's change the gear. So far I talk about the steady state, but now let me move to the oscillation. But before it, let me do the quick survey. If someone get a cancer, then we need to go get the chemotherapy. And chemotherapy typically happen 3 weeks interval 6 times. And if one go to the hospital, the nurse will ask, do you want to come next time morning or afternoon? Which one do you prefer? Let's make a vote. Who want to go in the morning? Hands up. Okay, and who wants to go in the afternoon? Okay, morning is more dominant. Let's see your choices, future. So we compare the morning group and afternoon treatment group. And this is an afternoon treatment group. And this is morning treatment group. So we tracking the 5-year survival rate. Afternoon group, 98%, only 2% people die. But morning group, 25% people die. That's a huge differences. And then another surprising thing is this difference only observed in the female. Male, no differences. Male don't care. Yeah. Female bodies, it looks like more sensitive than the male or more sophisticated or more upgraded. Yeah, I don't know. So, and then we wonder why. What differences come for this? And we found that your bone marrow activity, which make the white blood cell, are not constant. So bone marrow activity is very high in the morning compared to the afternoon or night. So what happens is when bone marrow activity is very active, if we give a chemo, then patients showed a very sensitive responses. Here, sensitive response means toxic responses. Then what if the patient showed the toxic responses, then medical doctor must have to reduce the dose level when they come to the next time. So that what we found is a female patient in the morning group, they got the dose reduction more frequently than a typical patient. But what about the male? I don't know why male bone marrow activity nearly the constant throughout the day. I don't know why. But anyway, only female has this rhythm, but male has very weak or non-detectable rhythm. That's why male doesn't care. So in summary, male, you go to the morning, and female, please go to in the afternoon. I think that's the optimal strategy at this point.
But Jay, I mean, the females do markedly better in the afternoon. Like the males' curves look like the female morning also, right?
So don't compare this directly. It's just like a normalized by the maximum level.
No.
Yeah, no, on the left.
Left? Sorry.
25% of the male die.
Oh,
About
Like,
that.
why does health treatment suck?
you drink and smoke and
Finally, a win for women.
yeah. I'm sorry. Yeah.
But also, I wonder, do the doctors do a worse job or the
For the male?
staff?
I don't
Oh,
know.
for the women.
Oh, yeah.
How do you know it's not effective the care that they're getting?
I think it could, of course, there could be some social environmental factor, but not only our study, there is some other study which did a random clinical trial. They also see the similar pattern. Only female has a difference, is male. No differences. We did it for the leukemia, but other study did a colon cancer, same pattern. So it's pretty universal.
Okay.
Yeah.
It's from the patient. It's not from.
Yeah, I think so.
Yeah. A nurse or something.
Then male should be also different, right? If the environment factor is really matter, right? So I really appreciate the male because they can be a control for our study. Okay. So anyway, our body is not a constant. It changes throughout the day. Morning, afternoon, evening, our bodies are not the same. We call this is circadian or 24-hour rhythm. Okay. Then how our body can generate this beautiful 24-hour rhythm is it's via the transcriptional negative feedback loop. So this is a cell. Inside of cell, there is a period gene. When clock and BML protein, activator protein bound to this gene, then these genes are activated so that per mRNA are transcribed, and then they are translated to the protein. So then we keep generating the protein, so we can see that the level of the per is now increasing inside of the cell. And then after 12 hours, they enter to the nucleus, and then they shut down their own transcription. Then no more production. So the increasing is now switch to the decreasing. So decay happened for 12 hours, and then there's no more per, right? And now we can resume the production. So this happened every day. 12-hour production phase, 12-hour decay phase. So your circadian clock generate the per protein 12 hours every day, and then they kill them for 12 hours. This happened every day. But this beautiful 24-hour rhythm basically tell the time to the rest of your body. Okay, so my per rhythms are actually originally in the Korean time zone. That's why first couple days of workshop, I looked very tired, right? Because of the jet lag. Okay, this is how that happened. But the question is, if you look at this, it's quite surprising how they turn on and turn off this transcription exactly at the same time, 12 hours every day.
Is this in all cells? Or?
Yeah, nearly all cells in your body. So every cell in your, even your skin cell has also circadian rhythm. So, so actually your cosmetic cream response also different depending on the time of day. Yes.
What does fix 12 hours?
Oh.
And it should be related to sun in some way, because 12 is, or 24 hours is just conventional. It can be 40 hours.
Oh, yeah. Thank you. Actually, this is I simplify for the explanation. Depending on the summer and winter day length, the ratio is also changed. But this 12 by 12 means like just maybe spring and fall.
But still there is feedback with the sun, right?
Yeah, exactly. But even you shut down the sun, this is maintained. If you go to the dark cage for a month, it's maintained for a month. Yes, you have a really clock in your cell. It's like this. Yes. Yeah, it's connected with the light, but it's also independent system, autonomous system. Cool? Yes, it's a very cool system. It's amazing system.
So it's an intrinsic oscillator.
Intrinsic oscillator, yes. Intrinsic oscillator, but it can be perturbed by the light-dark cycle so that we can adjust to the new time zone.
How is this intrinsic oscillator graded with human decision for 24 hours? Because it can be different.
Oh, yeah. Actually,
we can common numbers or whatever.
On average, it's 24, but there is some distribution, of course. So maybe J could be 24.2, mine could be 23.7. There is some distribution that determine the morning or evening type person. And if you want to know your circadian period, you go to the dark cave. If you get up earlier every day, that means your period is shorter than 24. Okay? But no one tested that so far, but
Just come to Edmonton in the winter.
yeah. Cool. Yeah. So this is a circadian system, but this is quite mystery. To do this, to make this autonomous system accurately, there are a couple of requirements. This protein need to enter the nucleus at the right time every day. Just imagine the cell, certain protein enter to the nucleus at the same time every day. That means all of you come to this room at the same time every day. Throughout 5 days, we failed, right? And then second, when they enter the nucleus, they should shut down the transcription perfectly. If you think about all the stochasticity or whatever, this is quite challenging problem. How they can achieve this? Let's look at the synthetic oscillator made by the synthetic biologist. Look at their oscillation. Do you think it's precise? No, we cannot use this as a clock, right? This is current hour level. But if we look at the circadian clock, this is a recording of the single cell in vitro for a month. Beautiful. So our nature clock is way better than our fancy synthetic oscillator. How we can achieve such beautiful, robust 24-hour autonomous limit cycle? Why they stop at the 50 days? Because they cannot measure anymore, because that's the limit of experiment. Cool, isn't it? So from now, I will talk about what is the difference between these two systems. Yeah. So typically, when we call repressor in the negative feedback loop means usually, to turn on the gene, RNA polymerase should bind, and then we need to turn on the gene. But repressor bind this position so that they shut down the transcription. This is a typical transcriptional repression mechanism. But let's look at it. This is a little bit weird. So repressor is protein, so that means the number of the repressor protein is usually 100 to the 1,000, but target is 1 gene. Do you like it? You're a stochastic model, so target is 1. Do you think it could be accurate or not? It's quite sensitive to the noise, right? Target is 1. It's very, but however, circadian clock utilize different way. Look at it. Repressor do not target the gene. They target the activator protein. So indirect repression. So what that means is repressor target the activator protein. Typically, number of the activator protein is 10 to the 3 or 10 to the 4. So rather than 1000 to the 1, they change the game to the 1000 to the 1000 regulation. I feel that they try to use the law of the large number thing here. Okay, so then what kind of indirect repression mechanism exists is there are 3 different types of indirect repression mechanism. Number 1 is blocking. What is blocking means? When activator is bound on the gene, repressor come to the activator on the gene so that they shut down the activity of activator. So we call this is blocking. And another thing is sequestration. Sequestration means repressor binds with the free activator so that it prevents the free activator go to the gene. And then third, displacement. Displacement means repressor binds to the activator and they eliminate the activator from the gene. So there are 3 different types of the indirect repression mechanism exists in the cell. Then my first question is which one is used in the circadian clock? Again, let's do the voting. Who think the blocking looks attractive? Okay, Jay saying like the blocking. Who think the sequestration? Cool, yes, stochastic people. Displacement. Okay, cool. You're all correct because all of them are used. Yeah, all of them are used. And then I checked the other oscillator like a P53 and NF-kappa B oscillator. They also use all of this. It's a little bit weird theoretically because at the beginning, because one of them is enough for the shutdown. But why use all these 3 together? It looks a little bit redundant. But in biology, redundancy means there is something. Okay, so we want to investigate this question, why all of them are used. So we begin with the just sequestration mechanism. Sequestration means repressor binds with the free activator and then they prevent activator go to the gene. Okay, it's like a American football. We block the attacker go to the borderline. So with this mechanism, we can get the very ultra-sensitive switch. So let's imagine if we have a more activator than repressor, let's say there is 100 activator, 10 repressor, then what happened? 10 and 10 are matched, but there are other free activator, they go to the gene and turn on the transcription. But if we have a more repressor than activator, then there's no free activator, right? So that shut down. And then that sharp transition happen around the repressor and activator levels are similar. So we can get a very good on and off switch system with the sequestration. And then now we performed some stochastic simulation with the Gillespie, but unexpected thing happened. So here yellow means we have a more repressor than activator, so that we expect that fully transcriptional repression, right? But when we perform the simulation, activation happened. Actually, this is not a transient thing because this is about 4 hours. That means our clock system will be completely messed up. Why? Why this happened? So let's imagine there are more repressors. Let's say there are 8 repressors, and we have only 1 activator. Usually they should be binding with one of this. But let's think about accidentally, with a low chance, they are released with this repressor, possible, right? And then they bind here with a very low chance. As long as they bound to here, then what happens is transcription will happen forever as long as he won't be there. Although this is an extreme event, as long as this happens, we cannot come back. They cross the border. Then how can we prevent this? I hate this. So key idea is we want to kill this one, right? If we add a blocking, then we can, this one will kill this one. So that if we add a blocking, you can see that that extreme event will be immediately restored to the original repression level. So if we have a blocking and sequestration, we can prevent this. So we are happy, right? But then what about activation phase? During the activation phase, we expect that it should be always on, but again, now, unexpected decreasing happen. Why? So this is the case when we have more activator than repressor. So usually repressor should be with the pre-activator, but accidentally, extreme event, they can go to the gene, then this shutdown happen forever. Okay, this is very low chance, but as long as it happen, it can mess up. Then how can we prevent it? I don't like it. Yes, displacement, pull off. Then if we add this, then again, this extreme event can be immediately restored to the normal. So basically, the circadian clock uses these three combinations to prevent this extreme event disaster. So indeed, if one of these mechanisms are mutated, you can see that peak-to-peak circadian period variation is become much higher, so that our oscillator becomes really unstable. Indeed, this three combination is critical. And then we did some calculation for the final factor. So if we have only sequestration, we have extremely high noise at this on pace. But if we add a blocking, then we can reduce here quite a lot, but payback we get increasing here. But if we add a displacement, we can get completely good one. But still here is a little bit sad. So hopefully someone can reduce this further. That would be great. So summary, direct regulation is, so far majority of the mathematical model for the gene regulation is focusing on the direct regulation. Repressor directly bound to the gene. With this one, I don't think we can achieve the robust regulation. 100 to the 1 ratio. But if we had an indirect regulation, now the game is 1,000 to the 1,000. And plus this combination, we can prevent the extreme event so that we can achieve the robust rhythm. So, so far I talk about how we can make this part is robust, but we still need one more requirement. To do this, they need to enter the nucleus at the right time every day. Oh, yes, question?
If you combine both direct and indirect repression, can you achieve the reduction in noise?
Oh, I never tried. Oh, I don't know. Hopefully that direct regulation can reduce that part. That would be cool. Yeah, I never thought about it. Yeah, thank you. Yes. So now more difficult mission is actually this. How can we make this 1,000 molecule enter the nucleus at the right of time per day? Oof. So Seokju did a very simple simulation, like an agent-based model simulation. We use the measure diffusion coefficient of the per molecule. We let the 100 molecule do the race from the cell membrane to the perinucleus. How long does it take? So we just performed some simulation with empty space or with some obstacle. So we can do it. And then let's look at the arrival time distribution. This. So arrival time distribution looks like this. That means certain molecule arrive to the perinucleus in a minute, but some arrive at 12 hours. So they do not arrive at the nucleus at the same time. Then question is, travel time of the per. That means your arrival time to this gate from the 0 to 12 hour of today. But how they enter to here at the same time every day all together. So, and what we found is, in collaboration with the Kyoung at the FSU, we found that they're waiting their friends. Can you see the ring structure here? So what that means is, like a Jinsu arrived 7 a.m. at the gate, but he waits for J because he haven't arrived. But J didn't enter here because Linji hasn't arrived. So everyone is waiting at the gate until all of your friends basically arrive. So they have very strong friendship, maybe better than us. So how they can achieve this? What is the mechanism underlying this collective behavior? So for this, we need to understand this simple thing. So for the repressor enter to the nucleus, what we found is they need to change the shape via phosphorylation. So original shape cannot enter to the nucleus. They need a phosphorylation to change the shape, and then they can enter to the nucleus. And that phosphorylation occurs in the cooperative manner. What that means is, first phosphorylation is very, very difficult, but follow-up phosphorylation is much easier. So first phosphorylation helped the second and third and fourth phosphorylation. We call this a cooperative mechanism in biochemistry. With this one, Daewoo found very interesting phenomenon. So what this graph is, this is key. So x-axis is in the cell. Let's make the very small circle, and then let's measure the concentration of the per in this small circle. Let me call that local per concentration. And then y-axis is in this small circle the fraction of the phosphorylated per protein which can enter the nucleus between the 0 to the 1, right? When local concentration is low, majority of them are unphosphorylated. And it's surprisingly maintained. But when it reaches to the certain threshold, suddenly switch up. Majority are phosphorylated, and then they enter the nucleus all together. So let me show this simulation. Someone arrived earlier at the perinucleus, but majority of them are in unphosphorylated status, so they cannot enter the nucleus because the level is still low. They wait more until more friends arrive so that they passing the threshold, that suddenly this unphosphorylated one switch it to the phosphorylated protein, and then they enter the nucleus together. So I'm sorry, it's not the friendship. They just need each other. So when all of you arrive this gate, then we got the key, and then we can enter to the nucleus. So indeed, with this phosphoswitch mechanism, although protein tribal time could be widely distributed, if we had that phosphoswitch, entrance time could be very narrowly regulated, and then we can maintain the circadian rhythm beautifully. So what is the simple idea underlying this? If we just focusing on just one molecule, then arrival time could be widely distributed, as you can see. But if we think about the arrival time of the 1,000 molecule average, because to get into the gate, we need 1,000 molecule. The main arrival time should be very narrowly regulated. Again, here is a law of large number exists. Circadian clock utilizes this idea very well. So, and furthermore, we did one more simulation. We add a more obstacle inside of cell so that we prevent that they are gathering at the perinucleus, then rhythm becomes very weak. Okay, so we wonder if this can be tested. How can we make your cell is more crowded? Yeah, one way is McDonald's add more fat inside of your cell so that we construct this is normal mice. There you can see that there's sleep-wake cycle is very regular. And you can see that even in the dark cage, well maintained, they get up a little bit earlier, right? 23.7 hour period, okay? But if we make them obesity, rhythms are gone. Okay, so you need to exercise, okay? But what else can cause the cytoplasmic congestion? And we found that aging, unfortunately, and autophagic malfunction or Alzheimer's disease, all of them are known to increasing the cytoplasmic congestion. Indeed, when we tested these three for the mice, indeed, their sleep-wake cycle become all unstable. Actually, same for the human as well. So kind of we suggest that this cytoplasmic congestion or traffic jam is very bad for our regular sleep cycle. So in summary, circadian clock achieve the robust rhythm because they target rather than one gene, they target the 10 to the 3 activator. And then they make the decision when they enter the nucleus not by the single molecule, but by 10 to the 3 or 10 to the 4 collective mechanism. That's why they can achieve this accuracy. Next step is, I didn't talk about this, how actually move toward to the nucleus, perinucleus. One summary is this. What we found is per protein don't use the microtubule to go to the perinucleus. They fully diffusion. But how they make the advection towards the nucleus is, what we found is in the cell, far away from the nucleus is very high diffusion. But here is a slow diffusion. This difference between diffusion then make the natural advection toward gradient towards the nucleus, but that is only true for the big molecule. Small molecule diffusion coefficient is same across the cell. Only big molecule are different, but per molecules are very big. So we found that this is a way make the natural toward to the perinucleus. Okay, so I have, I guess, 5 minutes more because I begin at 10 minutes late. Yeah, okay. So finally, I would like to talk about this. Noise is always bad. And let's begin with very simple, this negative feedback loop model. But we have a time delay here. How? This is time delay. So it is well known that if we include the time delay itself, it's always good for oscillation. It generates a beautiful oscillation. But rather than fix the time delay, let's think about, imagine the distribution of the time delay. Now let's increase the variance of the time delay while fixing the mean. Is it good or bad for the oscillation? Let's imagine we fix the mean of the time delay, but we're increasing the variance of this distribution. Does it mess up the oscillation or helping the oscillation? My guess was, of course, mess up the oscillation. But what we found is, as we're increasing the CV of the tau, oscillations are well maintained, but the shapes are changed. It reads the more sharper shape. Yeah, and then as a result, so that we measure the period and the amplitude and period sharpness, and the signal to noise ratio. And what we found is, even we increasing the distribution of CV of the tau, period is essentially constant. Amplitude is also constant, but sharpness of the peak is increasing. But this was very surprising. Signal to noise ratio is actually increasing. Of course, too much is bad, but certain amount of noise actually helping the signal to noise ratio is higher because it makes a sharper signal. And actually, not only that simple model, we checked the pretty common oscillator. It's a quite universal phenomenon. So sometimes noise can be helpful. So although today I talk about like a sort of basic sciences, but this research turns out quite useful. For instance, I talk about the phosphorylation regulated nucleus entry, right? Actually, Pfizer developed the compound which target that phosphorylation site, and then we collaborate together to develop a drug to change your circadian phases. Actually, it's successful because maintain the circadian rhythm is very helpful to prevent Alzheimer's disease. So that this drug was sold to the Biogen with a quite huge amount of money. I'm sorry, I didn't get anything from this money because I got the research funding. So yeah, that was a very good lesson for me. When you get the funding, that means when we sell something, there is no benefit for me. If you read the contract carefully, you will see all that kind of thing there. But recently, as Casso mentioned, our research for the circadian rhythm sleep are now adopted this new Samsung Galaxy Watch. So if you use this Samsung Galaxy Watch now, there is a personalized sleep schedule program. Underlying that, there is a differential equation and machine learning, etc. And this time now we got the money. Thanks for the lesson with the Pfizer. I knew what I have to do. So although I didn't talk about not much about the theory, there are several things you may help. Number one, this strong buffering structure idea. We only prove this only for the deterministic system. It maintained the steady state of deterministic system, but we don't know. Mean level of the stochastic system is maintained or noise level is blow up. We don't know. This is something interesting direction. And second, regarding this noise controller, actually this work only under the ergodicity, but we failed to prove the ergodicity. Hopefully you can prove it, then it would be beautiful. And then although we show that it can achieve the RPA, we don't know how quickly we can achieve it. Theoretically, it can take forever. We don't want. So that's another interesting problem, how to quickly achieve the RPA rather than just achieve it. And then finally, yes, we still fail to reduce this noise level. So maybe direct repression can be a source for this. Yes, that's another interesting question. So this is our group member. Hyunjung was here. So when he visited us during the summer. So this is a sort of first math bio center in Korea. So although majority are mathematicians, we do serve the real biological or medical problem. So if you have an interest in, please let me know. We have some short-term visiting, and then Jay Newby also our valuable visitor. And this is the funding for all this researching. And then this is what we are doing nowadays. Yeah, done. Thank you.
Yeah, with the adaptation, it seems like you were also maybe saying something interesting about identifiability from like stationary measured quantities, right? You're identifying areas where you can't really say anything about these individual parameters, right?
Yes. So kind of, if we translate this to the stochastic system is stationary distribution of the certain target molecule, how can we make them independent of the certain parameter? Of course, if we can write down, we can drive the closed form of the stationary distribution, then game is over. But usually we don't know, right? So then how can we still tell about that? That would be, and this for deterministic system, it's really quite a network topology property. So it would be interesting whether this network topology really determine the stationary distribution property as well. That would be very, I think, very cool, but very difficult, I think. To be frankly, Jinsu and we tried it. Have you served? Not yet?
Ergodicity?
No, left side.
No, so when everything is linear,
then of course,
they match, but not
non-linear, we still, we need a health. Yeah, we are happy to share what we have done. Yeah.
So the perfect adaptation, it seems to be sensitive to stoichiometry of the reaction.
Yeah, exactly.
And is it sensitive to the stoichiometry outside your
yes
group? Okay, so it is. Now, but many, if you look at this, something like this, right, there are many intermediate. Some of these reactions are sort of graded response.
Yes.
So it's maybe not exact. So how sensitive, the conditions that you had, the last condition had, what if I'm just a little bit off?
Actually, we don't know. So how much is it sensitive to the adding the more HER now?
Or a variation in the stoichiometry, a slight.
Or it's very sensitive because if you change the stoichiometry, then the V minus that equal to the 0 could be changed, right?
We change a lot.
Yes. So absolutely, you're correct. Anyway, this is whole depending on just stoichiometry. That's why reaction doesn't matter. That's the key idea of this study. Yes. Oh, yes.
So perfect adaptation was the return to the original level after the perturbation, but for your mean controller and noise controller, it looked like it never left. There was no perturbation in return. It just stayed. What's the reason?
Actually, if you look at carefully, they do. But I think my student choose a certain parameter looks very cool. But if you choose the other parameter, they go all the way and then coming down. So that's why we don't know how long does it take. The example we do, it looks like, but other parameter, it take quite a while. So, but this is a way more difficult problem because we only focusing on the steady state thing, but this is time problem. So I think you are the expert. So hopefully someone can serve it because RPA, this time is really all mystery.